Hello, everyone, and welcome to our online lecture series for plant biology. Today, we take a look at the other side of a plant, not the shoot and the materials that grow above ground, but rather the root and everything of the plant that's growing beneath the soil. Now, you might have some vague idea as to what exactly these different roles of the root are. But I will go ahead and say that different plants have roots for different purposes. Broadly speaking, root function can be classified as any number of these 5 different roles. Roots provide anchorage for plants, meaning they help keep it in place so that way the wind does not blow it away and the shoots are all of a sudden not facing the sun. Anchorage is very important because it allows the plant to orient its growth towards the sun, outcompeting neighbors for light. Absorption. In this case, the absorption of both water and nutrients. In this case, the nutrients are not the caloric, but rather the minerals that help the plant grow, because you can have things that provide calories, provide energy. Those are the foods that we eat that allow us to continue to move. And then in those foods that we eat, we have our vitamins and our minerals. In the plant's case, they're able to make their own vitamins, but they still need the minerals from which to build those vitamins. And so root absorption has to be of both water and minerals. Next up, roots can provide a way of clonal growth for a plant. So when seeds are not an option for you, because you happen to be the only colonist on a new deserted island, as a human that wouldn't work, but as a plant, roots can provide a means of clonally growing new individuals, and those new individuals will therefore open up the opportunity to occupy new environmental niches, so perhaps stabilizing the ground, or even perhaps providing new mating regimes. So, clonal growth is definitely one of the major functions of a root. We've also got conduction, so just as much as a plant must absorb both water and minerals through its roots, it has to conduct that water up. And then at the same time that it's bringing water up, oftentimes also bringing many of those, I'll put minerals down here, apologies. Not just the nutrients, but we can be specific, the minerals that need to go up the roots in order to, and up into the shoots to help metabolism happen. Sugar is oftentimes being transported down, as roots are an ultimate sink for sugars. We'll include more or a wider discussion about source-sink dynamics in a plant in a few lectures, but for now, just know that water's going up alongside those minerals that help the plant make all of the vitamins that it needs, and then sugars traveling down from the leaves into the roots, so that way the roots can continue to grow. And then last but not least, of great importance to us as humans, roots are storage organs. They are storage organs for those sugars that are traveling down. And it is from a lot of those storage organs that we derive our caloric needs as humans. I do love me a potato. Now, there's a couple other terms in terms of the root variation that we'll see. We notice that root architecture, or the way in which roots grow in different plants, is very, very different. Here on the left, we have a eudicot, so a plant with 2 seed leaves. Here you can think of something like a carrot or a rose. Eudicots will typically have a taproot, with a taproot being a very deep primary axis. Its goal is to get deep enough in the soil surface in order to get to the reserves of water far below. It's not that a eudicot does not have some lateral roots. You'll notice that the roots do spread, but compare the architecture of a taproot with these fibrous roots of a monocot. A monocot like corn. These roots are much, much more webbed. Lot of overlapping axes of growth, because rather than having one primary root apical meristem that can diverge and break off and separate, like we see in the taproot, so it has one primary axis of growth, and then all the offshoots from it. The adventitious roots, or the fibrous roots of monocots, will have many axes, and they will grow from these axes all in one concerted time, meaning that you get a lot more water absorption at the surface than you will below. And that's oftentimes the idea behind fibrous roots of monocots. They're going to absorb the water the moment it gets in to the soil at the surface, think corn, whereas a lot of taproots will not have as much growth at the surface, but they are instead looking for the deep reserves of water. And so we see these two various states of root architecture in our two major lineages of flowering plants. Now, when we take a look at taproots and fibrous roots and all of the soil attached, we therefore describe the rhizosphere. The rhizosphere is the volume, so all the material, of soil around the root. Because as you'll learn today, it is not just the root on its own digging its way through the dirt. The root will oftentimes secrete things into the soil around it in order to make that soil part of the habitat of the root, in order to make that soil amenable to the growth of the plant, in order to make that soil harbor the beneficial bacteria and fungi that a plant root needs in order to thrive. And so a rhizosphere does not just describe the root itself, but all of the soil that sticks to it, because there's a literal living connection between them. And you can see this observation fairly easy whenever you dig up a plant. When you dig up a plant, do the roots come out clean, or do they come out holding on to the soil? Obvious answer, right? The roots are holding on to the soil, not just because of the fact that they're adjacent to it, but because they're components of that root that have been added to the soil. And we'll be talking about what the root adds to the soil today. So again, we want to connect our discussion of the root apical meristem, which is where we're going to next, to our discussion of the shoot apical meristem. So as a reminder, in both the root and the shoot, we always have a region of cell division somewhere near the tip. In the case of our tunica corpus model of the SAM, you've got cell division occurring at the very growing tip. You've got elongation occurring behind that, helping cells grow long and propel the surface so that way the plant can grow tall. And then you've got a region of maturation behind that, where cells occupy their final function and mature into their three different plant tissues: the epidermis, the ground tissue, and our vascular tissue, comprised of both phloem and xylem. Xylem in the middle, phloem on the outside. Xylem, dead pipes; phloem, living sugar movers. Looking at this in our root, taking our previous lecture's drawing, we see that there is a slight difference in these three regions within our root apical meristem. You'll see that the region of cell division is not quite at the very tip. It is instead a little bit behind. After that, just like you'd expect in our shoot apical meristem, you've got a region of elongation and a region of maturation. So the further back from the tip you are, the more specialized your cells are in the root apical meristem, with the exception of this upfront area. So I'll go ahead and draw another drawing of our root tip. We have outside of it a colorized region that we'll refer to as our root cap. The root cap is that portion of the root ahead of the region of cell division. There is cell division occurring in this direction. So whereas in your shoot apical meristem, the tunica divided anticlinally, and the corpus, L3, divided periclinally, as well as some anticlinal division. So sending it back, in your root apical meristem, you are going to get division heading backwards after your region of cell division being held in just this small space. In many plants, it's something as small as being 2 cells. And then you also have separate cell division occurring ahead of that quiescent center. So I'll go ahead and label this now, quiescent center. You also get cell division forward, because as the root is digging through the soil, make no mistake, that's what the root cap is dealing with. It's dealing with dirt and rocks and stuff. There will be cell death. Lots of friction is happening at the root cap. And so the very, very tip of the root cap is your dead root cap. You can think of this as a football helmet, an American football helmet, that you're using to dig through the soil. Because in this case, your quiescent center is like your brain, is like your head. And you don't want to dig a hole with your face. You need a piece of machinery in front of it to help protect it. And that's the role of the dead root cap, to provide protection, to take a hit. Behind your dead root cap, you have your live root cap. And the live root cap has 2 distinct regions to it. The region that is living and copying and going in this direction, directly at the front, we call that the columella. And then as the columella either is pushed forward and dies and becomes the dead root cap on the side, or gets moved horizontally, it can become the lateral root cap. Both layers of your living tissue in the root cap. So the dead root cap is up front, protecting the columella and the lateral root cap. Behind the live root cap, the living portion of the root cap, we have our quiescent center, which is our region of controlling cell division. So you have these 2 cells that themselves do not divide. They stay stem cells in their current state forever. Forever. They are controlling the cell division of the cells around them. So I'll go ahead and actually, let's draw this in a smaller color here. The quiescent center, for some reason, ever since my first class, has been colored in pink usually. So I will color the quiescent center in pink, and then the region of cell division around it in blue. Ooh, that was crazy. Hadn't done that before. So you have the control of the cell division around the quiescent center. oftentimes those two cells in a plant root that control the cell division of the cells around them. So the quiescent center really is like the mastermind of this entire dance. It orchestrates which cells are dividing when and in which direction, and how frequently. Behind your quiescent center, you will get your regions of elongation and your region of maturation. The cells behind that quiescent center are therefore procambium. Early vascular tissue in the middle, or your ground meristem. Or if you're on the skin side of things, and you're in the region of elongation, we have our protoderm. Those being the three focused stem cell layers that make our three different plant tissues. So in your region of elongation, you have the protoderm, procambium, and ground meristem, that then in your region of maturation, way up on the root, or further back behind on the root, you have your specialized cells that have now adopted their true final form and function. So that is the basic architecture of our root in transverse section. But there are a couple new layers of tissue that are going to mature in your region of maturation. Things are not quite exactly the same as with our shoot. And think back to it. Our shoot is just supposed to perform photosynthesis. The root is not going to be performing photosynthesis, so it's not going to need leaf primordia. You'll notice I didn't include any discussion of any primordia happening here at our root apical meristem. So you have no leaf or root primordia. So what takes its place? What is the root doing, the root apical meristem doing, that's different from our shoot apical meristem? The first answer is all tied up with its function as a water filtration system. Not only does the root have to filter the water that a plant is taking in, because think about it. If I pour water onto a plant, can it decide whether it's going to drink that water, whether that water is going to interact with its roots, or does it itself have to filter the water? It has no ability to move. So the plant root not only has to be an absorber of water, but it has to serve as a method of filtration. But at the same time that it's filtering and bringing in that water, it can't be open to the soil and lose all of its water. So the plant root has two very specific dermal layers that provide protection and filtration for its water absorptive functions. Those two layers are either near the surface of the plant, the exodermis, or a subepidermal layer of the plant tissue. So it's going to be right underneath the skin, right underneath the epidermis, and it's going to have a specialized cell wall. And its specialized cell wall is going to allow nutrients, so the minerals and water in. Oops. But all of the pathogens and foreign things stay out. The bad stuff stays out. But this layer is not as filtration centric as our other cell layer in the root. If the exodermis is the outer first gateway entry, the endodermis, or inner skin, is the layer of cells directly outside of our vascular tissue. This is our permission slip into the veins of the plant. It is our filtering layer. And so let's take a look at the layout of our exodermis and our endodermis in some different plant roots. First and foremost, or rather in kind of arbitrary order, not first and foremost here, we have the root of a eudicot. And again, you'll notice that there are differences between our two-cotyledoned plant, things like roses, things like beans, and our monocot plants. The monocot plants, in terms of their stem, harkening back to last lecture, you can think of the monocots as having that vascular structure that does not have a pith and therefore cannot have secondary growth. Our eudicots do have a pith and can have secondary growth, and we might see that in the later developed roots. But for now, you'll notice that the general architecture of the root itself is different within this dicot and this monocot. The dicot has a cross pattern to its roots. The monocot is going to have a distributed ring. But otherwise, the endodermis and the epidermis and the exodermis are the same. So here, looking at our endodermis, you can see a very clearly defined circular ring. That is the endodermis of this plant root. Looking closer at the edge of the plant, you see the epidermis, a thin layer on the outside that's providing that cuticle, all of those waxy substances to help keep water in. But then additionally, you have an exodermis, a layer of cells that are expanded. These are part of the cortex, technically, so they arise from the cortex, but they are different than the cells behind them. Because they're directly adjacent to our epidermis, they've got very particular cell wall structures that allow them to do that primary or first pass filtration while also allowing the absorption of good things in and keeping bad things out. And think about how specialized that cell wall needs to be in order to allow water in, but prevent the stuff that's flowing in with the water that we do not want to come inside. In the same way as our exodermis and endodermis are defined bands in our dicot, we again see those defined bands in our monocot. The endodermis is the defined ring around the vascular cylinder, and the exodermis is the layer immediately underneath our epidermis. So you'll see again, cell layer on top, single cell layers are exodermis. You've got root hairs growing from the epidermis. These are epidermal extensions. Root hairs are not to be confused with lateral roots. Root hairs grow from the epidermis. Lateral roots, as we'll see in a little bit, actually grow from a layer inside of the vascular tissue. So they actually grow deep inside the tissue and extend their way out. And so you've got this additional exodermis and endodermis that's adding to our horizontal cross-section of our plant root. So just like we did with our plant stem, let's again dissect this plant root horizontally. You know that at the very outside, you have an epidermis with root hairs growing out of it. Underneath the epidermis, you will have the exodermis. Specialized cell layer that provides first pass at filtration and keeping pathogens or harmful things out. Beneath the exodermis, you have your cortex, because the cortex is what actually gave rise to our exodermis. Cortex is our living tissues. In cases above the ground, it's photosynthetic. It's performing a lot of the metabolism. It's doing a lot of the storage. It's parenchymal tissue. Inside of the cortex, you again have another filtering layer called your endodermis. In terms of filtration, the endodermis is much, much, much thicker and stronger than the exodermis. The endodermis is actually going to really put a hard stop to anything that isn't pure water from coming in. So you can get some suspect ions transporting into the cortex, but once they hit the endodermis, they're done. They cannot pass through the plant, cannot pass into the plant veins, because the plant does not want that traveling up to its leaves and killing the plant itself. So it will stay just in that subsection of the root, and only the most filtered, pure water and minerals will get put into the vascular cylinder of the plant. Now, interestingly, there's a non-filtering layer right underneath the endodermis. We have a living layer of ground tissue surrounding directly underneath the endodermis that's used metabolically for storage, for energy, for growth, called the pericycle. And it is from the pericycle that you're actually getting your lateral roots. So root hairs, epidermal extensions, singular cell structures. Lateral roots, they are internal to the root architecture. They come from the inside and literally eat their way out. So lateral roots have to digest some of the cortex, exodermis, and epidermis in order to make their way out and extend. Crazy cannibalism on an individual plant level. But that's how the organ forms. Now, beyond the pericycle, you'll have exactly what you expect. You have a layer of phloem on the outside. The phloem will be structured differently. As you can tell, sometimes it's surrounding completely. Like here, you pericycle and phloem directly smashed adjacent to one another. Monocots have very little pericycle, whereas here, the pericycle is much bigger. You've got phloem in these distinct bundles of 4, as I was drawing them, with the pericycle surrounding it. And inside of the phloem itself, you've got your xylem, which will oftentimes grow, particularly in our eudicots, in this kind of star shape, because the water pipes that grow early on are much smaller than the water pipes that grow later. So in your xylem, you actually have 2 different types of xylem. The early developing xylem, or protoxylem, and our later developing xylem, the metaxylem. And you can have vessels and tracheids of both of these. More oftentimes, you'll see a higher abundance of tracheids in your protoxylem, because your protoxylem is oftentimes your small stuff. And then your metaxylem is big. Once it's specialized, elongated, matured, the metaxylem will be giant. And you can even see how big some of these metaxylem vessel elements can get by looking at this monocot root. Look at those giant pipes for water. Compare them to some of the protoxylem that's in between as to how small they are. So you've got this subtle difference between early growing and late growing, components of your vascular tissue. And so, again, from the top, when we dissect a root, cutting from the outside in, we hit first the single layer epidermis, the 1 to 2 to 3 layer of exodermis, the cortex that's storing stuff, the endodermis that is doing a fantastic job of filtering materials, the pericycle that is living storage tissue in the vascular structure. It is from your pericycle that your lateral roots get the energy to grow. Inside of the pericycle, you have your food transporting phloem that's bringing sugar down into the roots. And you have your water transporting xylem that's carrying water from the roots up into the plant. Whew! So that is our root in cross-section, horizontally, and transverse section, vertically. We've covered the 2 different angles for this. So with the remainder of our time, we'll talk a little bit about some root variation and take a better look at what those lateral roots are doing. So here's an image from the textbook that neatly summarizes some of the other components that we've been talking about. You, of course, have the root cap, made up of both the dead on the outside and the living root cap, the columella being at the very front and center, and then the lateral root cap protecting behind. But of course, this is your football helmet that's helping the face of the root apical meristem, dig its way through the soil. As you go a little bit back, you will get a mucilage sheath. This is actually, whoa, did I forget to mention that earlier? I did. Outside of the root cap, you may encounter mucigel. So I'm glad I included this one, otherwise I would have forgotten one of the vocab terms. Ha. Mucigel is not present in all plants, but where it is present, it is incredibly important. It is secreted by the developing plant root, and these secretions are very much like mucus, like snot, because they actually contain the thickening agent that we use for jellies, something that you may have heard of called pectin. This will not be the last time. I think we mentioned pectin in an earlier lecture, and this will not be the last time we talk about pectin. Pectin is something that adds it's a thickening agent to things. So if your jam is too runny, then you add pectin, and all of a sudden, now your jam is turned into a nice jelly. Well, in addition to the pectin present in the mucigel, you've got a whole bunch of other digestible sugars in order to create this rhizosphere that allows other organisms, microorganisms, to grow around the root. And the mucigel is actually enabling the plant to cultivate, that's right, the plant is farming the bacteria that grow around the root apical meristem in order to keep this environment happy, in order to keep things around it that are healthy. Those healthy bacteria keep away the infectious bacteria, the bad bacteria that can be pathogenic. And at the same time, that mucigel can also help recruit fungi. The fungi will be incredibly important in a second once we're done discussing our lateral roots. So, going back to the diagram that reminded me of mucigel in the first place, you've got your mucigel towards the very tip, root hairs extending from that in your region of elongation, and you will get lateral roots forming in this region of elongation. This is when and where the lateral root is forming. Now, why? Why make lateral roots in the first place? Well, ultimately, the plant wants to have more surface area. Surface area is exactly what it sounds like. It is the mathematical term that y'all will have calculated from classes like geometry. Surface area is literally just the area on the surface of the organism. Now you want to have a whole bunch of surface area to allow your absorption to be maximized. The more surface area on the root, the more opportunity it has to absorb water. That's the major advantage of those fibrous root systems. Tons and tons of surface area adjacent to the surface of the soil, so that way they can absorb the water the moment it rains. Now, the lateral roots also allow for additional surface area by having the root grow sideways. These lateral roots, as I mentioned before, are actually outgrowths from the pericycle. Because the pericycle is still your ground tissue that's a little bit more meristematic. It has the ability to flex between different types of tissues. It's still got that boot print, that apical meristem boot print. It has not specialized into xylem or anything. It's got the ability to form a newly differentiating structure, a root that grows perpendicular to the current growth trajectory of the root. But it needs to be attached to the vascular cylinder. This is the reason why it emerges from the pericycle. Lateral roots are going to be directly plugged into the veins of the plant. If you want your water pipes to be connected to the old water pipes, you literally have to have a connection growing through them. So when we are growing our lateral roots, as these two images show, you will get the lateral roots forming from one edge of the pericycle. You can even see the extension here of the endodermis that is starting to grow in this emerging lateral root. And in this plant here, you've got an emerging lateral root going in each different direction. So these emerging lateral roots are now directly plugged into our vascular tissue. They're directly plugged into the veins of the plant and will be able to transport water and sugar quite rapidly. Now, the lateral root will initiate from the pericycle, and in particular, the pericycle that is next to protoxylem, those early small xylomatic tissues, and then it will eat its way through the cortex. It starts to digest down the cortex, which again, why do you want to eat the cortex to grow? Because it's a whole bunch of sugar and water and happy materials to grow. It is exactly what the plant needs in order to keep performing mitosis, and therefore the growing lateral root can utilize this store of energy to quickly push its way through the root system and make its way out the epidermis, where it will now become its own root apical meristem, growing horizontally for a bit. Now, we won't talk at all about the genes controlling this. There are some genes that we figured out, but I will admit that our understanding of lateral root formation genetically is not near where our shoot apical meristem understanding is. So that's one of the reasons why we're not actually including a detailed discussion of the genes here. But while we do know only some of the genes underlying this, we do know the hormone that signals for lateral root formation. And we will talk a lot about this hormone. It is auxin. So lateral root formation is auxin controlled. You may see auxin written as another abbreviation, or indole acetic acid. IAA is perhaps its other name, both used in the textbook and numerous Wikipedia pages. So you will see auxin, the hormone auxin, as IAA. And it's auxin that's controlling this lateral root formation. And so that's pretty cool. We can see some of the other different structures that we've discussed, a lateral root that's further up, an emerging current lateral root, the epidermis labeled here at the very, very outside. You've got the phloem labeled on the inside of the vascular tissue. You've got a cortex labeled. You've got protoxylem vessels, much, much smaller xylem vessels, and metaxylem vessels. Look how big that vessel element is there. Both same cell type, but whether you're built early or whether you're built late will determine how big that tissue will grow or that cell will grow. You've got the pericycle directly underneath. Ignore the passage cells, but you do notice that you've got a nice thick blue-stained endodermis here. So that kind of serves to review some of the different structures that we've discussed. So that, this is bothering me, bacteria, that is why we have lateral roots, to have more surface area and connect them directly to our pipes. There is one other structure that matters a ton when it comes to surface area. And the role of this cannot be overstated. In fact, for many different plant tissues, this structure actually has more surface area than the plant roots. This structure is actually doing much, much more of the water absorption than the plant root itself. And it's not even a plant. This structure is called mycorrhizae. Literally translated as fungal roots. Now, mycorrhizae are a symbiotic relationship. They're a symbiosis between fungi and plants. Fungi are really good at living in single cell layers. Because they are single cell layered, or good at growing as single cell layered, they maximize surface area. They're really good at digging their way through dirt and finding minerals and finding water. But they're really bad at producing sugars. They're not photosynthetic. Fungi are more closely related to animals than they are plants. That's truth. They are not photosynthetic, and therefore they need to find a way to get their sugar. Now, some fungi will break down dead things in order to derive their sugar. But other groups of fungi have developed the ability to trade. They've developed an economy of sorts. These fungi will grow into a plant cortex, so they will grow into plant tissues, establish themselves within a plant cell, so kind of like a trading post within the plant cell, and they will traffic in water and minerals, and in return, they get sugar. And in some organisms, some plants, the mycorrhizae are doing more of the water absorption than the plant roots are. So that really turns the whole narrative of a plant root is there to absorb water on its head. A plant root is there to grow in the ground, create a nice environment in the soil for different microorganisms to grow. And once those microorganisms have grown, they then need to form this symbiotic relationship so that way the roots can exchange water and minerals for the sugar that they have, the sugar that they've received from the shoots. And so you've got this really cute, small economy happening underneath the soil. And we actually believe that mycorrhizae were instrumental in plants even making it to land in the first place. Depending on which plant survey you look at, over 80% of all land plants form these symbiotic mycorrhizal relationships. So it's not like a small minority of plants doing this relationship with fungi, but rather the vast, vast majority of plants are utilizing this relationship with fungi in order to do their water absorption, which is pretty crazy to think about. And so that's where we have our lateral root discussion. maximizing surface area or increasing the amount of surface area that's there. And then mycorrhizae that take it even a step further, but they're not plant. They are actually fungal. They're not infectious. They've somehow come to an agreement here, and there's a lot of studying of the genetics underlying this, somehow come to agreement where the fungi will not just absorb the sugar. They will instead also provide the minerals and nutrients. Both groups are better when each works together rather than one stealing from the other. Take whatever life lessons you want from that. But both lateral roots and mycorrhizal roots are there to help increase surface area for more water and nutrient absorption. The very last thing that we'll talk about is root variation. Because we do get some different structures that y'all will be familiar with. Some root structures, and no, you are not looking at a potato, but rather a tuber of another plant that's also still forming this storage sugar, this sugar storage organ underneath the ground. A tuber is just a growth, a swelling of the stem. You can have stem tubers as well as root tubers. They are just there to store things. In particular, sugar. You can also get different types of roots in either the fibrous or the tap roots that are not tubers, but are instead propagative. They help proliferate the plant. You can have stolons, a stolon being an on top of ground, horizontal stem that helps the plant grow. And when I say on top of, it can be near the top, so you actually have this here, this structure here, as a stolon of this corm. And a corm is just a fleshy stem. So it's kind of like a tuber, but whereas a tuber has the opportunity for multiple apical meristems, so it's got multiple potentials for growth. Think eyes on a potato. A corm has a single apical meristem. So it's only got one place to grow out of. And that's the difference, the botanical difference between a tuber and a corm. Tuber, many apical meristems. Corm, singular apical meristem that it can grow from. Don't even get me started on bulbs. Yes, I understand there's a lot of technical differences botanically. So we've got tubers, storage organs of sugar. Corm, also storage organs, differences in the number of apical meristems. Stolons, they are on top of the ground or horizontal stems, roots that are adapted for propagation. That's spreading feature. Or, if your root is not above the ground in a stolon, the root can be below ground and a rhizome. These are your propagative roots that stay below ground. These are just a couple of the different examples that different plants have, with tubers, rhizomes, corms, and stolons being fairly common across the plant kingdom, so they get included into this lecture, to kind of allow for the plants to continue to grow without sexually reproducing. You'll notice that roots don't have flowers, but they're still able to propagate in case the above ground material is gone. So roots need to do it asexually by just breaking off pieces and growing, whereas shoots can do it sexually with flowers. And so that covers the vocab and the materials that we needed in terms of our root apical meristems. We now have a better understanding of the different root functions, whether it be anchorage, absorption, clonal growth, conduction, or storage. The two major different architectures of roots, taproots and fibrous roots, taproots in our eudicots and our fibrous roots in our monocots. We have an understanding of the three regions and the one root cap of our plant roots, in particular now, the additional understanding of our mucigel. We understand our quiescent center, the region of the root cap, the region of the root apical meristem that's not dividing, but playing the brain role for everything around, telling it when to divide. We understand the exodermis and endodermis, how those two different dermal layers help filter water for the plant. More on their cell structure later. And then we've, of course, got our discussion of all the different layers cutting from outside in onto our roots. So now we've discussed our two major apical meristems, the shoot and the root apical meristem. I'll see y'all next time for a discussion of some growth variation, so the different architectures of a plant. Think more some botanical innovations in terms of tubers and corms, but the other stuff that plants do. And then we'll discuss our last set of apical meristems, our secondary growth meristems. See y'all next time.